Good afternoon. I'm Brian Miller, CEO and co-founder of Sentien Biotechnologies. Thank you for the invitation from ARM to present again this year. I do recognize it's 5 o'clock. We're getting close. I'll try and be as dynamic as possible to keep people interested and enthused. So we are Sentien Biotechnologies. We are a cell-based company that has a technology that reprograms the blood via MSC secreted factors really to harness the potential of MSCs to restore balance to the immune system. We're focused initially on indications underpinned by systemic inflammatory response syndrome, initially in the acute setting. We are a clinical stage company with a phase 1/2 study in dialysis requiring acute kidney injury. I'll talk more about that in a little bit. And the key milestones that we've met, being a clinical stage company, we obviously have generated GMP material repeatedly. We have an open IND, obviously. But even in our clinical trial, data that we're not sharing yet, but we have completed the low-dose cohort for SBI-101, and we have some initial confidence around the PK side of things, and I can describe that a bit more as we go forward as well. So the problem that we're trying to solve is really a dysregulated response to injury, initially in a severe injury setting. So as a response to injury, a tissue injury, you have an immune-mediated inflammatory response. If not properly resolved, what you end up getting is some exacerbation of this injury and further tissue damage, either in the acute setting where you have a worsening of the inflammatory profile, or persistence in a more chronic setting where you have ongoing inflammation, ongoing progressive immunosuppression. What's really needed is a way to modulate that immune system, reset it back to baseline, and I don't have to tell this audience too much that cell therapies are the right approach to do that. In particular, MSCs have shown a lot of potential in this space because of their broad-acting, responsive, and dynamic properties. So with MSCs, sure where to point this. What's known about MSCs is expansive. They are broad-acting. There's a number of different factors that are being produced over time that collectively lead to this tissue repair and regenerative response. And what's really important with a cell therapy, they're also dynamically responsive. So in a setting where you have pro-inflammatory mediators like TNF-alpha, interferon gamma, IL-1 beta, MSCs will become activated, and they will produce a differentiated set of factors in response to injury. So it's really this dynamic responsiveness, someone referred to it this morning as a living drug, that enables a cell therapeutic approach in the setting of a very complex inflammatory disease like the ones we're treating. The challenge with MSCs has, of course, been a short half-life when injected. Presumably, or predominantly in the IV infusion, you don't get a really long exposure of these secreted therapeutic factors, leading to a relatively short and ineffective duration of activity. And that's really the problem that we set out to solve initially, was how do we enable sustained, durable, and controlled exposure of these MSC secreted factors to the blood. So the first instantiation of this solution set, sorry, I'm pressing the button here, there we go, is a cellular device where we're administering ex vivo the MSC secreted factors by enabling blood signaling to the MSCs to maintain that dynamic responsiveness and having the cells be protected in this extracorporeal device over time. So what that enables is a much longer duration of exposure. We've measured in the clinical setting, we're doing up to 24 hours of exposure continuously. In vitro, we've shown up to 8 days where the cells are producing factors. But it allows for bidirectional communication between the MSCs in the extracapillary space and the blood flowing through the inside of those hollow fibers through the membrane, which gives you that dynamic responsiveness I was referring to earlier. So what that gives us essentially is a number of things. We have significantly higher opportunities to dose cells. There's a longer period of cell viability relative to IV infusion. You get this broader therapeutic exposure I was describing. And a side benefit from the engineering of this is, if you can see, there's probably a laser pointer, there we are. These two blue caps represent ports that give us access to the extracellular space of the hollow fiber device. And what that means is we can start characterizing what these cells are producing in real time when exposed to patient blood. So it allows us to measure things on the PK/PD spectrum that people can't measure, obviously, when you infuse cells IV. So that data is going to be utilized as we start looking for incorporating PK/PD relationships going forward with respect to dosing, mechanism, potency development, et cetera. It's a really differentiating feature of our technology. Given that we have this cellular device, SBI-101, initially we are looking at clinically utilized blood circuits to create this as an add-on to that setting. So the types of applications we're talking about are dialysis, ECMO is extracorporeal membrane oxygenation for lung injury, photodynamic therapy, and apheresis. All these are existing clinically utilized blood circuits that give us access, if you will, to a bloodstream that we can integrate SBI-101 into to treat, again, what underlying are a lot of systemic inflammatory disorders. So this represents approximately 1 million patients per year. Where we are starting is on the top with dialysis, in particular in the ICU setting, dialysis requiring acute kidney injury. These patients are very highly inflamed. Gives us a good opportunity to again explore the pharmacology of this product in a phase 1/2 setting. It is the single most expensive diagnosis among hospital inpatients, so it's a very severe need. There's not been really any change for these patients in decades, so it's an ongoing problem where both patients and providers are very interested in finding a new solution. Mortality rates are very high as well, but it is an opportunity for us to integrate with standard of care for these patients. So we are currently engaged in a phase 1/2 study. What we're showing here on the left is a little bit of preclinical data that we had in an efficacy model of acute kidney injury. This model was a dog model where we took out one kidney and on the remaining kidney did a 90-minute clamp of the vasculature. Normally people do approximately 45 minutes of the clamp. Once that clamp was released, we waited a full 24 hours before treatment, mimicking more like what you would see in an actual clinical setting. Again, normal for these types of models is to do a 45-minute clamp. Maybe you wait an hour to administer where you're trying to prevent this inflammation. Our approach really was to get full-blown ischemic reperfusion, AKI. We had a 4-fold increase across all groups in creatinine. So very significant AKI injury upon initiation of treatment. After that 24-hour hold time, we then exposed the animals to a 12-hour treatment. So what that meant was we set them up on a dialysis circuit for 12 hours. We took away the product and then measured the 7-day survival as a function of that. So we had 3 groups. The negative control was fluids only for the animals, 50% survival. Dialysis with an acellular control had 70% survival, and SBI-101 had 91% survival. And again, I want to highlight what we viewed as evidence of this, this blood reprogramming concept, where we're exposing the animal's blood, in this case, for 12 hours of treatment, taking away the product and having a durable response out to 7 days. So that's the type of thing we'll be looking for as well in the clinic. Our clinical trial the overview is on the right-hand side. It's double-blind, randomized, controlled. We have a low dose and a high dose at 2 different cell masses, in each case exposing the subject to 24 hours of treatment. We are measuring, obviously, safety as a first-in-human, looking at renal-specific efficacy, but really as we want to understand the pharmacology, again, it's this PK/PD sampling from the product, correlating that with different changes to the blood that we think is going to help us start to elucidate mechanistically what's going on. Interestingly enough, from the preliminary readouts that we've had, and we hope to have an interim readout towards the end of this year, and hopefully the full readout at this conference next year, we have shown that by sampling from that extracapillary device, or the port, we know that the cells are producing factors over 24 hours, and the pattern of MSC secretion profiling is consistent with a dynamic response. And what I mean by that is we see cytokines that after a few hours are increasing in their expression level in the presence of inflammation, both in vitro as well as in these patients. And in the absence of those inflammatory stimuli, we see a different pattern. So the patterns that we're seeing clinically, again, are consistent with what we've observed in our own hands with a dynamic response of these MSCs. So early on, we're very encouraged that the product it seems to be working the way we intended, the way it was designed, where you're getting this broad-acting dynamic response in the clinical setting. And we're encouraged to see what the rest of the data will look like as it becomes available. This is the beginning for us. So on the left-hand side, we're talking about patients that are in an ICU setting. They're getting continuous blood perfusion over days upon days to treat their critical illness. Where we see this going is expanding more into chronic applications. Initially, step by step, getting into a plasmapheresis environment where you have shorter but perhaps more frequent duration of treatment. For diseases where they might be on the IVIG bandwagon, for example. Moving further out, getting into what we call offline treatments, where we might take some blood, condition it offline, and then give it back to the patient. And the furthest out on the right, where you're conditioning cells ex vivo for a certain inflammatory signal, and then collecting the secretome effluent and giving that essentially as a drug. Where we think we can compete is in moving down this line. Again, it's this ability to deconstruct the interaction of two living systems, these MSCs dynamically responding to blood, that can enable insights that will allow us to move down this pathway. So we're excited to be in this area where there's a tremendous amount of unmet need. We also see tremendous market opportunities as we move further to the right as well going forward. So the team that we have, I will highlight. I will not say anything about myself. We've just announced this week the hiring of Dr. Pedro Huertas as our Chief Medical Officer. He's an expert in translational medicine with experience in big pharma, small biotech, and cell therapy companies. We're thrilled to have him, as well as our scientific co-founder, Dr. Biju Parakadin, and Chris Jamiti, whom many of you know at this conference. We've attracted a world-class scientific advisory board, including some of the leading nephrologists, as AKI is our first indication, but also people that are familiar with translational medicine from Pfizer days on the bottom, and Dr. Greg Ruzati from Celgene in the cell and tissue engineering space. So really, in summary, our therapeutic focus is going after diseases that have this complex systemic inflammatory profile that monotherapies are really unable to treat. The platform is enabling MSC-secreted factors to condition blood, create this immunoregulatory, immunomodulatory response. We're looking at initially, as I mentioned, programs in the acute space, but then eventually expanding out once we have more data into the autoimmune and chronic disease spaces as well. So we have a clinical trial going in AKID. Again, we hope to read that out. Interim results by the end of this year. And then we'll have the full readout sometime in the middle of next year. Again, expanding into other programs in the acute on chronic and chronic space. With the stage of the company that we're at, we're pleased with the way we've been de-risking the technology going forward. We do feel like we've hit enough milestones that we are asking the core hypothesis to our company is, can this alternative way of administering MSC-secreted factors have a clinically relevant response? And we feel confident that question is being asked in the clinic, and we're excited to look at the results. So I thank you for your time, and I'm happy to take any questions offline afterwards. Thank you.